Oh, hi, everybody. Chapter 7 is our next topic, which is the skeletal system. We've actually started to talk about bone already when we discussed tissues, and we examined compact bone in lab recently when we looked at the osteons. So a bit of this introductory part of the chapter you've already heard of. So when we think of the skeletal system, of course, we think about its role in providing support to our body. The fact that our nervous system, both the brain and spinal cord, are protected by the skull and vertebrae, respectively. And the fact, as we'll talk about in an upcoming chapter, our muscles, specifically skeletal muscle, is connected to our bone, of which we have conscious control over. This is, of course, different from smooth or cardiac muscle, which is involuntary. And the fourth major function that we will also talk about in this chapter is the fact that when you look at the matrix of bone, which is, of course, very strong due to the calcium present in the collagen, our bone gets its strength from calcium to some extent, but also we use calcium in a variety of different physiologic processes, ranging from muscle contraction to blood clot formation to, of course, building strong bony matrix. And so we store our calcium in our bone matrix. And when we need more, there are mechanisms available to extract a bit of that calcium from the bone bank, if you will, in the matrix, and use it for whatever the body needs to use it for. And I've given you again a couple examples. Muscle contraction, blood clot formation, both require calcium. And then we also take calcium, of course, from the foods that we eat, and we replenish that which we've used from the bank, and we put that calcium back in the bank. And we're going to talk about cells that are instrumental in helping us bank that calcium. So we're always able, the body's always able to extract calcium and use it, but at the same time, replenish what it's taken away and bank it back into the formation of more bony matrix. Let's talk a little bit about the various types of bones based upon their shape. There's four basic terms we're going to talk about. A lot of these are very kind of intuitive and very self-explanatory. Like this first one, of course, called long bone. And of course, we think about bones like the femur. Can you think of any other long bones of the body? Maybe you're thinking about the humerus, the ulna, the radius, the tibia, the fibula. Now, these are all examples of long bones. And hopefully you've begun to learn those for lab. On the surface of our long bones, specifically at the ends of our long bones, we find cartilage, shown here in white. This is hyaline cartilage. The reason that we use this term articular is simply to indicate that this cartilage is at an articulation or a joint. So the hip joint would be up here. The knee joint, of course, would be on the distal end of the bone, in the case of the femur. Short bones. These generally have equal lengths and even widths. And when we think of short bones, we should consider most of the bones of the wrists and the ankles, because they're this type. This happens to be a bone of the ankle, one of the bones that we'll be actually learning about coming up in lab very shortly, called the talus. well, this is your basically your heel, part of your heel. Actually, it's located above the heel bone, but it articulates with what you would generically call the heel. Short bones. Flat, well, you all have a good sense of that, having just recently learned about the skull bones, or you will if you haven't. The parietal bone, you know there are two of those, or will be learning the fact that there are two of those. They make up the vast majority of the superior and lateral parts of the skull. And again, the name is very appropriate. They are indeed very flat. We're going to talk more about those. And of course, what kind of structures do we have here along the sides? Okay, those of course are sutures, where one skull bone meets another skull bone. Irregular. These come in a variety of shapes. But when I think of irregular bones, I think of the vertebrae. I also think of many of the facial bones. The sphenoid of the skull, that's a pretty irregular bone. The zygomatic, the vomer, these are just very bizarrely shaped bones. And actually, we can go and talk about a fifth category called sesamoid or round bones. It's sort of a special type of short bone, actually. And when we think of the sesamoid bone or round bone, most of the time I think of the patella or kneecap. It's an example of a sesamoid bone. There are other ones too, but this is the most common one. There are not a lot, but just think of patella when you think of sesamoid. Think of sesame seed, maybe. It's kind of got that shape. By the way, these particular shapes are described in one of the lab exercises. So you will have already read about this, or you will soon be reading about that coming up shortly. Likewise, that same exercise in the lab manual talks about long bone structure. So we want to take a few moments today and talk about some of the structures that are found in a long bone, like the femur. That's of course what we're looking at here. And this particular femur has been both longitudinally sectioned, the top half, as you can see here, and we also see a bit of a cross section. So this is a really nice way of depicting some of these anatomic terms. We've already talked about the fact that we have articular cartilage on the ends, and now we're going to use a term to describe the ends of long bones. This is referred to as the epiphysis. If it's SIS, that's one. If it's SES, that's plural. So we have a proximal epiphysis of the femur, that which is closest to the trunk. Okay, and remember the hip is up here, hip joint, and then we have the knee joint down below, which would be defined as the distal epiphysis. And then we have this very long shaft portion called the diaphysis. So a proximal epiphysis, a distal epiphysis, and a diaphysis. In recent editions, the book has also defined what they refer to as the metaphysis, which is sort of where the epiphysis ends and the diaphysis begins. And that's all we're going to really say about that. We don't often talk a whole lot about the metaphysis regions unless we're talking about specific anatomical structures that would be, of course, found in that particular area. But again, we don't use that particular anatomic location or talk much about it other than to define it right here. Now notice the presence of compact bone here, mostly in the diaphysis region. Now remember, this we saw before in lab when we examined the osteon, the microscopic analysis of the cross-section of human dry ground bone is what the slide label was. I don't know if you remember, but I did mention this is called compact bone. And you can again sort of see it in this cross-sectional view, the majority of the diaphysis here is compact. You can see it coming down here. The other part or type of bone is called spongy or cancellous. And you can see where it gets the name spongy because it sort of looks like a sponge. And you'll notice that in the epiphysis here, in the proximal epiphysis, and also actually in the distal epiphysis, we find the same thing. And that is a lot of spongy bone or cancellous bone. Now, there is a little bit of compact here in the epiphysis. You can kind of see me tracing it here on the very outer edge or periphery of the bone, but the majority of the epiphysis is spongy. As we get down into the diaphysis, though, look what happens. The extent of spongy begins to sort of decrease or become reduced, whereby then the compact bone becomes the more common type of bone, say in the diaphysis. And as we'll talk about a little bit later, but I might as well mention it because I've obviously defined it here, there are these bony branching plates here of the spongy bone called trabeculae. Okay, trabeculae. Those are the bony branching plates. When we look in the spongy bone of a long bone in the epiphyses, we'd find what's called red marrow. You're probably familiar with red bone marrow. It has actually a liquid consistency to it, a rather viscous liquid consistency. It's not a solid, it is a liquid. And we often talk about or hear about bone marrow transplants, right? In the case of leukemia, where they extract marrow from a donor and give it to a recipient, right? So they're extracting the bone marrow by typically injecting a needle into a bone that has a lot of spongy bone, and they literally will then extract the liquid marrow, yeah, and then transfuse that into the leukemia patient. Because the red bone marrow is the site of particular types of stem cells that give rise to red blood cells, white blood cells, and platelets. The formed elements of blood come from what are called hematopoietic stem cells in the red bone marrow, and we'll talk more about that when we get into the blood chapter. We've already talked about the articular cartilage, the fact that it's hyaline, but gets the name articular because it's at an articulation or a joint. When we look at the surface of a long bone, which would really be all surfaces except where you have the articular cartilage. Okay, so where the articular cartilage ends, like where my pointer is, and then extending down to the other end, except where there's articular cartilage, the surface of your bone is made up of a membrane called the periosteum. Now, you don't see that in the skeleton or in the bone that we're going to be examining in lab coming up soon. That has been since dissolved away when they prepared the skeleton or prepared the bones for manipulation in the laboratory. But in you and I, we'd have this rather thin, tough, vascular covering of dense connective tissue. Now, vascular means what? Vascular. Vascular means that there are blood vessels present in this membrane. And we'll talk later about the role that it plays, but obviously you can see here it talks about how bone formation and repair occur thanks to the presence of this vascular membrane, which is going to be providing particular types of cells that are going to produce bone for us. It's called the periosteum. When we look back into the diaphysis, we noted earlier the presence of compact bone just under the periosteum, but also we noted that there's a bit of spongy here around the very inner part of what's called the medullary cavity. So in the center of a long bone, in the diaphysis of a long bone, You have this cavity, which contains both blood vessels, as you can see here, the blue and the red denoting veins and arteries respectively, and also a different kind of marrow called yellow bone marrow. Now remember, we had red bone marrow in the spongy bone of the epiphyses, and now we have a different kind of marrow called yellow marrow that's found in the medullary cavity of the diaphysis. And the primary role of the red marrow is in storing fat. That's why it looks yellow. Now, yellow bone marrow is rather interesting because if for any reason there's a need to make more red bone marrow for whatever reason, this marrow has the ability to differentiate into red bone marrow, but its primary function is in storing fat. The last thing I want to mention on this slide is the fact that when you look at the inner lining of the medullary cavity, okay, it would be right next to the spongy bone. So you've got periosteum, compact bone, then a little bit of spongy, then you have this membrane that lines the medullary cavity called the endosteum. So don't confuse periosteum with endosteum. Endosteum is lining the inner medullary cavity. And we will talk more about the role it plays a little bit later in helping to make bone. So the periosteum and the endosteum are pretty darn important membranes. This is simply a review slide where we've taken the terms in the last 4 or 5 slides and just obviously thrown them up here. I thought breaking them down slide by slide would make it a little more palatable, but if you want a good review of anatomical structures of long bone, this is the slide to go to. Here we're looking at a cross-section through the femur. And again, just to orient you, on the surface here would be the periosteum. It appears to have been removed, but that's that fibrous vascular membrane. Note the presence of a lot of compact bone here. This is where we have the osteons, the microscopic structural functional units of compact bone, osteons. And then, of course, we see the bony trabeculae of the cancellous or spongy bone with a medullary cavity here in the very, very center, which, of course, you know, is basically this space. Now, that's not to say that spongy bone doesn't have osteocytes, because it does. But with respect to the osteons, those structures that we said look like a bunch of trees that have been kind of sectioned in the woods with the rings emanating from them and the bone cells in the lacunae, remember all that? We're going to review that in just a moment. That's what we've got out here. But in the trabeculae, as we can see from this inserted sketch, if you cut through one of those little trabeculae like we've done here, we would still find osteocytes, okay, in lacunae, but the orientation of those cells is not around a central osteonic canal like it is in compact bone. We still have lamellae, which we'll talk more about in a few minutes, but that's basically the collagen and the calcium that makes up sort of the rings of the tree in the osteon. Well, we sort of have ring-like structures here too in the cross-section of the trabeculae. Here we're looking at a cross-section, or actually a longitudinal section of the proximal end of the femur. This would be near, again, the hip joint. Note that the articular cartilage has been dissolved away. This is, again, from a skeleton, as has the periosteum. And all this is really trying to point out is the extent, the extensive extent of spongy bone here in the proximal epiphysis. The same thing would be found in the distal epiphysis of a long bone like the femur. We have a thin layer of compact, but most of this is spongy. Now, the other structure that we'll be talking about a little bit later when we discuss ossification is something called the epiphyseal plate. Another term for that is growth plate. Now, this says remnant, which indicates that this bone is done elongating. Had this been cartilage, this bone would have the potential to continue to grow or elongate. But because this individual is probably older was older than 25 or 30, the growth was done in this individual. And so we see what once was cartilage has been replaced here with bone, and we can actually see that in this cutaway view. We'd also be able to see that in an X-ray, for example. Here we're looking at a flat bone like the skull, and just to sort of orient you and point out some differences here in a flat bone versus, say, a long bone. Obviously the outside and inside where the brain would be. In the case of a skull bone or flat bone, note the presence of compact here as well as here, with spongy sandwiched in between the compact. You can see the trabeculae here of the spongy or cancellous bone if you look close. Okay, we're going to take a chunk of the diaphysis and blow this up. And this, again, is review. We've talked about this quite a bit recently in lab. But when we look in the compact bone, as I said, we have microscopic osteons. And I won't bother to go over all of the anatomy. We've talked about this before, but you should certainly review that. Remember the osteocytes in the lacunae. We can see the concentric rings called lamellae that extend out from the center. In this longitudinal section, we see several osteonic or central canals, and the fact again that they contain blood vessels and nerves. The nerve is in yellow, blood vessels obviously in red and blue. But one thing that it also points out quite nicely is how we have interconnections, if you will. These are called Volkmann's canals or perforating canals that basically bridge or link the central canals of the osteons. Now again, this would be a microscopic structure, but it's a hollowed out part of the bone which contains, of course, blood vessels and nerves. And so that's sort of how blood vessels and nerves can extend out into the compact bone, delivering oxygen and nutrients and so forth to the cells. And of course, the waste products can move back eventually to larger blood vessels here in the medullary cavity. Because remember, there's no blood in the osteon other than in the central canal. No blood out here, only in the central canal. We've talked again about the presence of osteocytes in lacunae, one osteocyte per lacuna. We've already talked about these concentric rings, what look like rings of a tree. These are called lamellae. It's basically the extracellular matrix, because remember, this is technically a tissue. And extending through the lamellae are these small, minute tubes called canaliculi. Within each canaliculus are cytoplasmic extensions of the bone cells, which we can see here. So here's an osteocyte, here's an osteocyte, here's an osteocyte. And it's through those little canaliculi that extensions of the cytoplasm of the bone cell are found. And it allows, remember, for rapid diffusion of nutrients from the blood supply here in the central canal all throughout the entire osteon. So nutrients can get to these cells via diffusion, and waste products can move from all these cells eventually back and be removed via a vein. This is simply a magnified electron micrograph that shows one osteon, of course, the central canal. Here's a few of those cavities or lacunae, within which would be a bone cell or osteocyte. We can see these rings of lamellae. If we look kind of close, we can see those. And we can also denote or see the small little canaliculi here, those minute little tubes through the lamellae, which contain the cytoplasmic extensions of the bone cells that reside inside the lacunae. Very, very nice photograph. Remember again, the lamellae are composed primarily of collagen, which remember is one of the types of fibers of connective tissues, super strong. That in concert with the calcium that we said is stored in the bone, in the lamellae, provides a very rigid, strong trait to bone. Simply another colored electron micrograph showing the blood vessels in the central canal, and again, the lacuna here, these spaces which would contain the bone cells. This is a fascinating photograph that shows one single osteocyte within its lacuna, and we can actually see some of the cytoplasmic extensions running into these minute tubes of the lamellae. It sort of reminds me of a Stegosaurus dinosaur. Remember the bony plates on the back of the dinosaur? That's what it reminds me of. Okay, we want to talk a little bit about how bone is formed in our bodies. And there's two basic mechanisms. One is called intramembranous ossification, and the other is called endochondral ossification. When we talk about how the skull bones are formed, so the frontal bone, the parietal bone, the temporal bone, the occipital bone, as well as the mandible and the maxilla, and even the clavicle, and some of the facial bones, it's via this method called intramembranous ossification. And I've given you here a hyperlink to a really good YouTube video that I'd like you to watch as you review the steps that we're going to talk more about in just a few moments. So watch that video, refer to the part of the PowerPoint that talks about intramembranous ossification, and that's just a really, really excellent tutorial that will clearly lay out the steps. And I want you to note that this process, whereby most of the flat bones of the skull are formed, along with a couple other specific bones in the face, area of the face, this is the primary mechanism by which bone forms during the first 2 months of fetal development. Compare that to the second type of ossification called endochondral, which takes over sort of where intramembranous leaves off. So from month 2 through actually early 20s, this is the primary method of how bones form. And again, I've given you another hyperlink to a really good YouTube that will take you through the steps that we're going to be talking about in a few minutes here. They're both excellent, excellent videos. And then finally at the bottom is a third hyperlink which you can watch that reviews both kind of together. Okay, so let's talk about intramembranous ossification. If you look at that word intramembranous, what does that mean? Intra means within. Inter means between. And students always get those two prefixes mixed up. So again, intra, within, inter, between. So this process of how the future bones of the skull, the clavicles, some of the facial bones, including the mandible, the maxilla, and the zygomatic, are formed as a result of this process. And it begins in sheets of what are called mesenchymal tissues. These are embryonic tissues that are found in layers or sheets. So if you go back to that that slide I showed you a few moments ago, think about the frontal bone. Think about the parietal and temporal occipital bones of the skull. So before those were fully bony, they were made up of sheets of mesenchymal tissue, very thin sheets. And there comes a point in time when these mesenchymal cells will change or differentiate into another cell type called the osteoblast. Osteoblasts build bone. So just think of the word B, or the letter B, osteoblast. It's a close cousin to osteocyte, actually. But these cells, osteoblasts, are going to make or build bone. And one of the first things that happens once those mesenchymal cells differentiate into osteoblasts is these osteoblasts will begin to lay down a bony matrix. They'll begin to lay down calcium and other compounds that will begin to calcify in those various layers or sheets. And something called a primary ossification center begins. And it's from here that the bone will radiate out. The formation of the bone will radiate out from between those sheets. Now, once the osteoblasts, which again produced that initial matrix that became calcified, when those osteoblasts find themselves surrounded and trapped by the matrix they just secreted, they become osteocytes. So they change their role. They've done their work. They've laid down the bony matrix. Now the osteoblasts become true bone cells or osteocytes. And again, this begins at a primary ossification center, and eventually other little islands of developing bone will form. Eventually these islands, these primary ossification centers will fuse together, and what once was sheets of vascular mesenchymal embryonic tissue now become bone. So that original connective tissue, that mesenchymal tissue, is altered radically and now becomes bony tissue. And if one looks at the inner and outer portions, let's say, of a skull bone, like the parietal bone or the frontal bone, we have an outer periosteum and an inner endosteum. If you think back to that slide I showed you earlier of the skull bone, where we had, remember, the compact bone on the outside and the inside, and the spongy in the middle, sandwiched between the two? Well, now you can begin to perhaps see how that initial bone that was laid down, which was actually spongy, has compact bone deposited over the surface of it and on the outside of it. So there's initially spongy bone produced, and then we have compact bone laid out from that, and also inward from that, to create, again, that sandwich, if you will, of spongy in between compact. Again, go back and look at that photograph that I showed you earlier of the cutaway view of the skull bone. This is a series of sketches that sort of walk you through some of the steps that we just talked about. I think also I would definitely go back and watch that YouTube video and stop it, pause it, as you review these various steps. And I think intramembranous ossification will be much more understandable. But just take your time, go through that YouTube video and refer to these steps. The other major process by which bones are formed is through endochondral ossification. And this pretty much would be 98% of the bones in your body are produced via this mechanism. So we don't start with sheets of mesenchymal tissue. but rather we start with hyaline cartilage. And you might remember me mentioning when we did cartilage in lab, I mentioned where we find hyaline cartilage, and one of those places was the fetal skeleton. Before you had a bony framework, most of the bones in your body when you were a fetus were made of hyaline cartilage that become replaced by bone. Again, I'll remind you, there is that hyperlink to a video that you should watch when we get done talking about this mechanism and refer to it. So let's pretend we have in the fetus what will become the future femur. We'll call it a cartilaginous model. This is hyaline cartilage. So we've got chondrocytes in lacunae, just like we said we find in hyaline cartilage. Well, there will come a point in time, starting about month 2 of fetal development, where some of these chondrocytes in the future diaphysis of the femur, and again, we'll use that as an example, but this applies to most all the long bones. Those cartilage cells will begin to enlarge in size. And the matrix that surrounds them begins to calcify. So what you see denoted here is this yellow material. That's the calcification that's beginning to occur between the lacunae, which house the cartilage cells. Now, as this calcification process continues, the cartilage cells or chondrocytes have no way of getting nutrients, and they begin to die. At the same time that this calcification is beginning to occur here in the center of the future diaphysis, there is the formation of what's referred to as a collar, a thin collar of bone that forms around the outer surface here of what once was cartilage. Now, we've talked about osteoblasts earlier. Remember what they did? Osteoblasts build bone, they make bone, they lay down matrix. And that's what's going on here. The osteoblasts are laying down a thin collar of compact bone. Now remember, this is a longitudinal section. You have to think about this in the three-dimensional manner. So we were just seeing a portion of it. The bony collar is going to extend around the front and also around the back here. So just like a napkin ring, you know, holds the napkin when you go to a fancy restaurant, we're starting to lay down a bony collar around this structure, whose whose matrix here is beginning to calcify, and these chondrocytes are beginning to die off. They enlarge and die. But we're starting to form this early periosteum around the future diaphysis. Now remember, our cartilage cells have died off in the middle, in the future diaphysis of the bone. And what we begin to see now is that blood vessels begin to invade from the periosteum. We already formed that as that initial bony napkin ring collar, right? So blood vessels begin to invade into those regions that were occupied by chondrocytes, which have, remember, enlarged and died. And that influx of blood vessels will carry osteoblasts inward, and those osteoblasts are going to occupy where the chondrocytes used to be. And in the same way that the osteoblasts laid down spongy bone in intramembranous ossification, so too will these osteoblasts begin to lay down bone here in the future diaphysis. When the osteoblasts find themselves surrounded by bony matrix that they've in turn produced, they become osteocytes. This process begins again in the center of this future femur in what's called the primary ossification center. Primary meaning first. So the ossification occurs first here in the diaphysis region with the initial death of the chondrocytes and then the invasion of osteoblasts that start to lay down bone. The spongy bone you can begin to see forming here. And we notice again compact bone forming out from it. And then we'd have a periosteum here on the very outer edge. Because remember, this is a longitudinal section. Now, we have a third type of bone cell that we want to talk about. So we've already discussed osteocytes, bone cells. We've talked about osteoblasts, another type of bone cell, but this one builds bone. And now we have a third one called the osteoclast. Osteoclasts cleave or cut or break down bony matrix. So I use the term cleave to help me remember what this C stands for, like a meat cleaver, cuts meat. Well, this cell cleaves or breaks down bone matrix. You might wonder, well, why would we want to do that? Well, remember, what do we eventually have to have here in the center of the diaphysis? We have to have a medullary cavity, right? So to get a medullary cavity, you've got to get in there and start to degrade the calcified matrix that was there originally. And so those osteoclasts will invade, just like the osteoblast invaded from the outside via blood vessels, and they will begin to break down that bony matrix and create a cavity, which later becomes the medullary cavity. So they're very, very important in that process of creating the medullary cavity. Now look what's starting to happen here in the epiphyses. This is denoting the enlargement of cartilage cells, because remember, this is all still cartilage here in white. This is brown bone. So we're seeing that same process that occurred in the diaphysis begin to occur in the epiphyses. That is the enlargement of the chondrocytes and some initial calcification of that matrix. Now, I hope you know what I'm going to tell you next, and that is in the epiphyses, those chondrocytes are going to die. We're going to have invasion of blood vessels that will carry osteoblasts that will now begin to produce bony matrix, just like they did earlier in the central part, in the medullary cavity region. But now we have ossification occurring in the epiphyses in what are called secondary ossification centers. So this is a secondary ossification center. Here's another one, and here's a third one. So the cartilage cells die, the osteoblasts come in, they lay down bony matrix. When they're surrounded by bony matrix, they're called osteocytes. When the baby is born, this is what his or her femur and many of the long bones probably looks like. Meaning we have some ossification that's already occurred, but we also have quite a bit of cartilage still present, hyaline cartilage shown here again in white. During infancy, during the first few years of life into early childhood, look what still is present in the future femur. Well, we have the secondary ossification centers obviously present. We have the diaphysis present, but we still have hyaline cartilage in what are called epiphyseal plates, or some people would refer to them as growth plates. So growth plates or epiphyseal plates are still hyaline cartilage. As long as you have epiphyseal cartilaginous growth plates, this bone can continue to lengthen. And of course, as the child grows, he or she gets taller, right? We all know that. That's because of the presence of epiphyseal growth plates. Now, there will come a point in time which, for you and me, was in our early to mid 20s, when all of the long bones become fully ossified. In other words, the growth plates go away, they transition from cartilage into bone. Literally what's going on actually as the two ossification centers fully meet and we're done lengthening, and we're done growing in height. And so this, of course, is the typical femur you'd find, say, in an adult. Note again the presence of the remnants of the growth plates. We talked about that earlier. So this is telling me where the growth plate last was, but it's not cartilage anymore. It's now bone. And of course, we see all these other structures that we alluded to earlier. The medullary cavity. The presence still, however, of articular cartilage on the epiphyses. Don't forget about that. That doesn't go away. You still always have that. So if this does not become bone, this stays cartilage, hyaline cartilage. And so these are the various steps, starting with the cartilaginous model in the fetus and working our way all the way up through child and adolescent years. So this is sort of fetal stages. Here's adolescent years. And then here we are in the adult, in you and me. Now, let's take a few moments and talk a little bit about the epiphyseal plate or growth plate. So what I'm talking about now is I'm describing this structure in a 13-year-old, let's say, or this structure, or this structure. This partial, well, not partial, it's fully cartilaginous. So let's think about the proximal epiphysis region of a femur. So we're talking about these regions. So here we are on the epiphysis of a long bone, call it the femur if you want. We have the secondary ossification center up here. We have the primary ossification center somewhere below my arrow. And this is that piece of hyaline cartilage that we defined as the epiphyseal or growth plate. It's still cartilaginous. And there are 4 basic zones noted here. What is of most interest is this zone number 2, okay? So it's just under what we call the resting or reserve cartilage. And again, to orient you, don't lose sight of where we are. Secondary ossification center off the screen up here, primary down below, off the screen down below. This second zone of proliferating hyaline cartilage, it says here mitosis. You know what that is. That's active cell division. So here the cartilage cells are undergoing prophase, metaphase, anaphase, telophase, parent cells giving rise to daughter cells, blah, 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 blah. So the cartilage is undergoing cell division. As long as you have this zone of proliferating hyaline cartilage, you will continue to have lengthening of the long bone. Now let's look what lies beneath that second zone. We have actually 2 zones. We have this zone of hypertrophic cartilage, and you can take a look at how these cells are a little bit larger in size, they're closer together, right? That's also denoted here in this histologic stain section. We can see that these cells, these chondrocytes are getting closer together, they're getting bigger in size. The matrix region between them is thinning. There isn't much there. There's a little bit here, but there isn't very much. These lacunae are getting like right next to one another, aren't they? There was quite a bit of matrix up here in zone number 2 and zone number 1. So hypertrophic means the cells are getting bigger, the chondrocytes are getting bigger. And then below that, in zone number 4, look what's happening. The cartilage is calcifying. And when we start to calcify the matrix, what happens to the chondrocytes? We said this earlier. When the chondrocytes are surrounded by matrix, they no longer have a means to get nutrients or get rid of waste products. So these chondrocytes die off. And they eventually are replaced by osteoblasts, which build bone. And that's what's going on down here. The osteoblasts have come in, they've begun to secrete the calcium salts that are going to be needed to make the strong bony matrix. These are blood vessels here. So as long as you have, again, the presence of this growth plate and you're actively dividing the hyaline cartilage, which will eventually be replaced by bone, the bone will lengthen. But when you get to be in your early 20s, the secondary center and the primary centers eventually fuse and the cartilage goes away completely and the bone is done growing. And we see the remnant, say in an x-ray or in a cutaway view across a longitudinal section of a bone, we can actually see that too. This is an x-ray of a child, and we're looking here at, this appears to be a posterior view of, I think it's the right, yeah, the right knee. And the reason I know that is I can see the fibula here. What the arrows, of course, are denoting is the presence of the growth plate. This black structure is still indicating the presence of hyaline cartilage. Now, this space right here, we're going to talk more about this later, but this is the fibrocartilage where we find the lateral and medial menisci found in the knee joint. This is not hyaline cartilage, so just ignore this for right now. But this should have a gap here. This is not bone, it's fibrocartilage to cushion the two bones. This is indicating the presence of an intact growth plate, so this kid's still growing in terms of his or her length. And of course, this final bottom end of the slide is showing this timetable that I think is really, really interesting, that kind of talks about, you know, when do these different processes of endochondral ossification occur. Now, you don't have to memorize this table, although we did talk a little bit about how intramembranous was primarily at work during the first 2 months of fetal development, and then from month 2 on, endochondral, you know, began. But I think it's interesting to note that a couple things to note. One is that, you know, even into your early 20s, you still have ossification occurring. In other words, replacement of mesenchymal tissue or replacement of cartilage, hyaline cartilage. So by the time you're 23, if you're a woman, or 25 if you're a guy, at that point you're done growing. All the bones have been fully ossified. Now you might note that in females, it occurs a few years earlier than in guys. And that's because of the effect of estrogen that we'll talk a little bit more about coming up on another module. Okay, I think I'm gonna end it right here. Yep, I'm gonna end it here, and we'll pick up where we left off in the next module. So get into chapter 7, start reading. I basically covered the first 5 or 6, 7 pages of the chapter.